Good evening and welcome to another episode of Garden Hour, brought to you each week by SDSU Extension with a panelist of experts. I'm going to be your host tonight, and with us tonight we have Madalyn Shires. Dr. Shires is our extension plant pathologist with the Department of Agronomy, Horticulture, and Plant Science. And we also have with us Connor Ruen, who is a graduate student with our department, and he's going to be sharing with you some of the work that he's doing for his graduate projects. So we'll start off this evening with Maddie, and what are you going to be sharing with us tonight?
Tonight, I'm going to be talking about some common viruses and phytoplasmas that you might see in your garden or in your home garden, like lawn areas.
Sounds good. I'm going to be talking a little bit tonight about one of the issues we see in raspberries sometimes, which are viral problems. So you can chime in on that also. And Connor, just in the brief 1 or 2 sentence, what is your project about?
Today I'm going to be talking about soil health and why it matters, and my project is in living mulch studies.
Sounds great. So we'll go ahead and get started, and Maddie, you can take it away.
Okay, let me get my screen shared. All right. Hopefully everyone can see that. All right, so like I said, tonight I'm going to be talking about some common viruses and phytoplasmas. Most of these are found sometimes in South Dakota. I worked with roses in my PhD, so I thought I would share a picture of my favorite rose. It's a Daisy's purple. So yeah, we'll get started. Tonight we're going to talk about viruses to start with. This is a list of the different viruses I'm going to talk about, and I'll show you some pictures, and then we'll go into a couple of things about phytoplasmas. Tomato spotted wilt virus. Most of you have probably heard me talk about this if you were on Garden Hour last month in June. So tomato spot wilt virus is a virus that is mostly found on tomatoes. It is spread through thrip feeding on infected plants, and then those same thrips going and feeding on healthy plants. Again, tomatoes are mostly the only thing affected by this virus. It does seem that we see more tomatoes affected on the western side of South Dakota than on the eastern side, but we have seen it in both sides. So it is something to definitely keep an eye out for. It does spread very rapidly through, especially in greenhouse settings. So it's very important to make sure you monitor for it. And I'll show you some pictures here in a minute. If you do have tomato spotted wilt virus, there's a few management practices that you can do. One of those is to utilize resistant varieties. Again, if you've ever dealt with viruses, you will probably know that there is no chemical treatment once a virus is in a plant. So another great management strategy is to remove symptomatic plants. That will keep the thrips from feeding on those infected plants and spreading the disease. You can also consider chemicals to reduce thrip numbers. However, if you've ever dealt with thrips, or if you were in South Dakota in May of this year, you'll know that thrips are kind of hard to control. So it's not practical to think that you will eliminate all the thrips, but you can take use some chemicals and some other strategies to reduce those numbers. This here is an example of tomato spotted wilt virus. These all are pictures from Rhoda Burrows' fact sheet. You can kind of see the ring spots that are here on the leaves, and then you can also see the ring spots that are here on the fruit. And you can even see it on the unripe fruit along with the ripe fruit. So it's pretty noticeable and pretty distinctive. And again, if you see this, you want to remove those plants because it does spread very, very easily and very quickly. The next virus is tobacco rattle virus. This is a common garden and ornamentals virus. There are many plants affected. Some of the more common ones are tulips, marigolds, daffodils, beans, peppers, and tomatoes. So you can see it affects both our vegetables and our ornamentals. This virus is transmitted by stubby root nematodes. If you've never heard of a nematode before, they are a small worm that lives in the soil and feeds on plants. So that's how this virus is moved. A nematode feeds on an infected plant and then feeds on a healthy plant and moves that virus. Management, again, is similar, and it's going to be similar on all of these. Always do good scouting, look for symptoms, make sure that you use resistant plants if you do have tobacco rattle virus that pops up in your gardens frequently. Some resistant plant examples are phlox and zinnias. Remove infected plants, including the root system, if you do see this virus. The virus likes to stay in the root system. So if you don't take those roots out, those nematodes can still feed on the infected plant roots and spread the virus. So it's really important to make sure you dig the whole plant out if you see symptoms. And here's some common symptoms. We actually received, the reason I'm talking about this virus is we received a submission of these pictures here through the garden hotline, and this was on some peonies in the Pierre area. So it causes ring spots similar to other viruses. It also causes odd mosaic or that weird yellowing that you're seeing here. So those are kind of the most common symptoms. And again, if you see this in your plants, like all viruses, there is no treatment for it, and it's best to remove the plants to keep it from spreading to others. Cucumber mosaic virus. If you receive the garden newsletter that Christine puts out every couple of weeks, you might have seen the write-up that Christine, Rhoda, and I, and one of Christine's students did. So this virus affects a wide range of vegetables, fruits, bedding flowers, weedy plants, and ornamentals. This virus is transmitted through aphid feeding. It can also be transmitted through mechanical transmission, which is where leaves touch other leaves. So another reason that it's really important to remove your infected plants. It can also be transmitted through seed. So it's important to not save seed from plants that have had CMV. Management, again, is to remove affected plants as soon as you see symptoms. Plant resistant varieties whenever possible. There are some out there. CMV can also be hosted by weeds. So it's really important to manage your weeds that are in around, garden beds or ornamental beds or greenhouses, because they can serve as a reservoir of the virus. Also, because this is a mechanically transmitted virus, it's extremely important. It's always good practice, but it's extremely important to make sure that you clean all tools that you use whenever messing with, removing plants, trimming plants, whatever that have cucumber mosaic virus, and you can clean those tools using alcohol or bleach. But very important to make sure you do that, because it will, cutting a infected plant and then cutting on a non-infected plant will spread this virus. Here's some examples, and these came from that earlier. This came from a fact sheet. I don't remember which one. I have it in my references at the end. But as the virus name suggests, it is a mosaic type virus. So you get the abnormal yellowing that starts on the leaves, and it kind of gives this mottled effect. It can also affect fruit and gives, again, kind of a mottling effect. So yeah, that's CMV. Tobacco ring spot virus is another one that does pop up here and there in South Dakota. It infects many garden plants, including cowpeas, soybeans, grapes, and blueberries. Like the tobacco rattle virus, this virus is spread through dagger nematodes, and it can also be spread through plant propagation from infected plants. So if you're taking cuttings off of blueberries or grapes, that can spread and will spread this virus. Management is like many of our other viruses: remove infected plants, use really good cleaning practices with your tools between plants, again with the alcohol or bleach. If you are planting back in an area with a previous very heavy infection of tobacco ring spot virus, you could also have a soil test done to look for those dagger nematodes. Not many people do that because usually if you've removed those infected plants, it's not necessary, but it is an option out there for management. And here's some examples. Again, you can kind of get some of these, chlorotic ring spot looking symptoms. Also produces a oak leaf pattern. And that's not seen just on tobacco, that is seen on some other types of plants. And that's this here is a really good example of that. Our next one is High Plains wheat mosaic virus. The host of this is sweet corn, field corn, wheat, grassy plants. It is a very common virus throughout the Great Plains. It does pop up here and there. This virus is vectored by an eriophyte mite, which is a very, very tiny mite. You can barely see it with your naked eye. This virus can also be spread through seed as well. It is very problematic in sweet corn. So it's really good to kind of keep an eye out for it. Management is to remove symptomatic plants and do not save seed if you have observed any infected plants. Also management of grassy weeds, and you can utilize crop rotation if possible in your garden. And here's some examples of High Plains wheat mosaic virus on sweet corn. So it kind of produces these chlorotic yellowed leaves, but this is also kind of a hallmark of this virus. It used to be called maize red stripe virus, and that is for a very good reason. It produces these red stripes on the leaves. So if you ever see your sweet corn looking a little bit like either of these examples, it's possible you have High Plains wheat mosaic virus in your crop. Another virus that does pop up from time to time is Impatiens necrotic ring spot virus. There are several hosts of this virus. Some of the most commons are impatiens, petunias, begonias, cucumbers, tomatoes, and peppers. This virus is spread through thrips feeding, which is the same type of vector as that tomato spotted wilt virus that I started with. Tomato spotted wilt virus and Impatiens necrotic ring spot virus are actually in the same family of viruses, so they behave similarly. Management of this, just like all of our others, make sure you clean your tools really well, remove infected plants, and do not perform any sort of plant propagation off of symptomatic plants. And here are some examples of that. Here again, it makes ring spot. That's the reason virus names are really fun. They usually have the name of the most common symptom. So you can see these here. All right, another one that I'm going to talk about, and I am actually talking about this because I did my PhD on this virus, and it does pop up here in South Dakota sometimes. Rose rosette virus. This virus is an Amara virus, which means it is in the same family as the High Plains wheat mosaic virus that I talked about a couple of slides ago. Roses are the only known host, and it is both wild roses and your ornamental landscape roses. This virus is spread through the feeding of eriophyte mites, similarly to High Plains wheat mosaic virus. It can also be spread through plant propagation and through seeds. So you can see that they're very similar in behavior. Management, just like everything else, is to remove infected plants, don't make cuttings from infected plants, and to do really good practices of cleaning on your tools. If you've never seen rose rosette virus before, I consider you very lucky. It is a plague of a plant disease in the South. I did my PhD at Texas A&M. So we will see our roses start doing things like this. So this rose has made a witch's broom where it's growing multiple stems out of a cane. It's also producing pink thorns, which is abnormal for roses. And if you can kind of tell in this picture, the canes are actually very thick, which is not normal. So it's a big problem in the South, but it actually started in Manitoba, Canada. So it has spread from Canada all the way down to Texas and all across the East Coast. I have a few more pictures. This is our most common site in Texas. If you've ever planted a knockout rose, you know it shouldn't look like this. And so you'll be going around parking lots and down highways, and you'll see roses that just look like this here. This abnormal reddening and abnormal leaf shapes, I kind of have a close-up of that. We've got a big witch's broom here where we've got tons of stems all kind of coming out of one node on the plant. We've got abnormal thorn amounts, we've got crinkled leaves. So this is it's a big problem in the South and the East Coast. And like I said, it can be seen in South Dakota. It's just not extremely common, but it is one that I got to work with. And so I thought I would talk to you a little bit about it. Okay, now I just have a couple of phytoplasma diseases. So if you've never heard of a phytoplasma before, it is a bacteria that lives a virus similar lifestyle. So it is limited to the phloem or the nutrition pathways of the plant, which is like how viruses are. This bacteria does not grow on the outside of the plant. It has to be in the phloem. Typical symptoms of phytoplasmas are witch's brooms, where I talked about that's where you have a lot of stems coming out of one node. Yellowing and chlorosis is another common symptom, and abnormal fruit production or abnormal bloom production is another very common one. How do phytoplasmas move from plant to plant? Leaf hoppers are your most common vector, but phytoplasmas can also be moved in seed and through plant propagation. The reason I was bringing this up is someone had asked about Aster yellows disease the last time I did garden hours, so I thought I would include it. So some of the more common phytoplasmas, there's several others, but these are just a couple that I wanted to talk about, is Aster yellows and X disease, or also known as little cherry disease. So this is an example of Aster yellows. I use a sunflower because most of us love sunflowers and know kind of what they should look like. You can see that this bloom head is very deformed, misshapen. There's petals coming out of the center of the bloom. And that's kind of similar here. You can also see that this never matured up and it's very abnormal. So this is very common symptoms of this Aster yellows disease or Aster yellows phytoplasma. Aster yellows phytoplasma can be found on any type of crop. It can be found in wheat, in zinnias, marigolds, any type of aster flower or a grassy crop, it can cause problems in. And you have to remove symptomatic plants because leaf hoppers are very common. There is no way to control or manage leaf hoppers. So if you do have this pop up in your gardens or in your yard, it's best to remove all symptomatic plants to reduce the disease load to prevent the leaf hoppers from spreading that disease. This next one is X disease. X disease is kind of near and dear to me. I worked on that after grad school in Washington. So we most commonly see that here in South Dakota on choke cherries. Many of us know and love choke cherries for many different reasons and uses. So choke cherries will start showing these symptoms in the late summer into the early fall, and that is this abnormal reddening on the plant. It may also have this bunchy witch's broom growth that you can kind of see starting here. The fruit may also look abnormal. What I worked on was in sweet cherries, and this is what we see in our sweet cherries whenever we have X disease, this pale, very small, misshapen fruit doesn't ever mature. Many times the fruit over here, while that does look like a normal standard cherry, it actually doesn't taste very good. So the phytoplasma affects the taste and the sugar transport. As a plug for some research I have going on, if any of you have choke cherries that look like they have X disease in the fall, please reach out to me because we would love to get some samples. We have a nationwide survey going on of different phytoplasma diseases. And we would love to be able to have some samples from South Dakota. So with that, here's all my resources. If you would like to check it out, feel free. And that is the end.
Thank you, Manny. I have one question for you. I often get this question when we talk about viruses. Is it okay to compost the plants that you remove, or do they need to be burned, or?
It's really going to depend on the virus, because you could very easily have nematodes in your compost pile. So, you know, those nematodes, they're not going to be afraid of compost. They may even be attracted to it. So if you have viruses that are being spread through nematodes or something like that, you do take the risk of them feeding on, you know, that plant material before the virus has died and spreading it still. Whenever we deal with viruses, we always recommend that people dig out the entire plant, including the roots, because viruses do love to live in the plant roots, and bag that up and just throw it away into your normal trash. It's usually just the safest measure, because especially if you don't know exactly what virus you're dealing with, you take a chance of reinfecting your yard whenever you were trying to get rid of it.
I had a second question on that, and it just flitted through my mind.
Well, if you remember at
Yep.
the end, we can cover it again.
Right, yeah. So, Connor, we're eager to hear about your project.
Can you see my screen?
Not yet.
How about now?
Yes.
So my name is Connor Ruen. I am a grad student here at South Dakota State University, working underneath Dr. Christine Lang and Dr. Tandina Leia. And today I'm going to talk about soil health and why it matters. So just to start it off, what is soil health? SARE, which is the Sustainable Agriculture Research and Education Center. It defines soil health, or healthy soil occurs when biological, chemical, and physical conditions are optimal. So I'm going to talk a little bit how you can optimize your soil health in your garden or lawn, wherever you see fit. So biological properties, that includes biodiversity, such as plants, insects, animals, and microfauna, or micro and macrofauna, as well as pest pressure. So plants obviously could be your grass or any plants you are growing, including weeds. Then insects are pretty straightforward. And then micro and macrofauna, that's more of your earthworms and any small mites that you might not be able to see. And then microfauna is the super small stuff you need a microscope to see. And how to kind of manage the biological properties. The best way to manage the properties is crop rotation. That is just rotating through the different plant families that we have. And you can see with this chart below the different crop rotations that you can commonly use. And then also include flowers that can attract beneficial insects that can help you prevent pests and any disease or pest issues that you may have. And then also add aspects of permaculture. Another property of the soil is chemical properties, and that includes pH, optimal nutrition, and the reduced use and free of harmful chemicals. And some of the ways that you can manage soil properties is through the use of soil testing, and this can determine if your soil is acidic or basic, and then also if you have any nutritional deficiencies or overabundances. Then also form a fertility plan for your garden, and you have several resources such as the Garden Hotline, which is on SD State Extension, or you can email an extension agent directly, ask a local master gardener, or even you can just use Google, which has all kinds of resources to look at. Then I wanted to include this because it kind of is interesting to see how different pHs in your soil can affect how available or how plant available different nutritional sources are in the soil. So as you may have heard before, the kind of Goldilocks zone of pH in soil is anywhere from 6 to 7 for pH. And that's because, as you can see in that range, all of the major nutritional components of soil are most available at those points, which is shown by the wider bars compared to the narrow ends on either side of the spectrum. The last and third property of soil is the physical property. That includes soil structure, compaction, aeration and porosity, texture, and bulk density. All these have an effect on how the soil absorbs water, allows roots to grow through, and also can determine whether you should plant carrots or if you should plant something that is a little more water tolerant and doesn't mind having its feet wet. How to manage these physical properties, maximize living roots in the soil. This helps increase the aeration of the soil just by adding macropores as those roots die off and regrow and press through different compaction layers. And then also reducing tillage and passes over the soil. This helps prevent compaction and also hard pans, which is a layer of compacted soil that's usually right below where you are tilling. Then cover crops or covering soil. This helps hold the soil in place as well as prevent compaction. And this can be done in your walkways by having clovers or grasses that are easy to manage. And then the biggest thing with physical properties is time. It's one of those things where it's very easy to screw up years of work in an afternoon if the time says so. So it's definitely important to be patient with the physical properties and not to try and rush anything. So for my project, I am looking a little bit into the soil physical properties. So here is a penetrometer, which is an instrument we push into the soil, and it measures how much force it is necessary for the tip of that penetrometer to go through the soil. And that measures it in PSI. So I also found that corn, they can push through compaction layers of up to 408 PSI with their root tips, or at least that's the measured water capacity that they can push through with PSI. So it's very important to understand that a compaction layer can limit how deep your plant roots can go and also limit how much space they have, as well as if they can absorb the water that may be lower in the soil profile. And as you can see, the penetrometer, it just gives you a reading. So I pushed it into the ground and it just states if it's low, medium, or high. And as you can see in this reading, it was 650 PSI for me to push the penetrometer about a foot into the soil. So that shows that there's likely a pretty heavily compacted layer underneath the soil somewhere. I'm also looking at water infiltration, and this is a measure of how quickly the water can be absorbed by the soil. And this effect is very well correlated with compaction, porosity, and the high salt levels. So if you have very high saline or sodic soil, you are very likely to have very slow draining water. And that can be measured very easily with infiltration rings, as shown in this picture. And to use the infiltration ring, we put a 10-inch PVC pipe on the ground with a 2x4 over it and use a rubber mallet to pound it into the ground. It's definitely easier when it's just rained compared to right now. When we did it today, it was pretty hard to get the PVC down into the soil. Then you put a layer of plastic. So in this picture, we just use a grocery bag from Walmart, and that works just fine. And that's done to prevent you from mixing up the soil when you pour water in, since mixing up the soil can cause it to basically clog some of those micro pores and kind of sway the data on how much will be penetrating the soil. And then using 1,000 milliliter graduated cylinders, we pour in the water every minute. And then we look at the graduated cylinder, and as you can see this image, there's about, I think it's 450-ish milliliters in the first or so minute. Then we refill the graduated cylinders, and then we just kind of sit around and wait. And we do that for about half an hour. And then after half an hour, if the infiltration rates have kind of equalized and we have pretty similar rates, we stop the reading and go to the next one. So if you have any questions, this is my contact information, and my resources were at the bottom of the slides. If you have anything you're curious about, feel free to contact me at my phone number or email.
Thanks, Connor. A nice overview of some of the different research methods that we use to come up with some of the information that we share with you over time. I see that while Connor was speaking, Amanda Bachman was able to join us. You know that our panelists are really dedicated when they rush in from traveling on the road from their job and join us anyway. So
yeah, I'm pulling the John Ball and presenting from a hotel room. But I did want to try to make it on this week since I missed everybody last week with the hailstorm in Pierre, which was very exciting. And I now have a new windshield on my car. So fingers crossed that there is no severe weather while I am on the road again this time, because my car is again exposed to the elements. But I did want to hop on and share a couple of the insects that I've been seeing out and about, and also some upcoming events that we have. The first one here is a Dogbane beetle, and this is just one of the really cool beetles that we have in South Dakota and the Midwest. I actually took a picture of this one when I was in Nebraska for the North Central Sustainable Ag Research and Education State Coordinators meeting. So we got to see some cool urban gardens, and I was out there chasing around some very pretty insects. So you can see that the Dogbane beetle, it kind of has that same body shape as a little bit like a ladybug, maybe a little bit like a Colorado potato beetle. It is in that Chrysomelidae, that leaf beetle family. So it does kind of look similar to some of the other insects you might be seeing in your garden, but it is very much a metallic green beetle. It's metallic green all over its body. And I included it because it might be something that you may confuse with another insect that is not so sort of great to have around. Dogbane is, like the name suggests, it feeds on dogbane, which is in, related to the milkweed. So it's another one of those plants that has a milky sap. So if you have a patch of dogbane, odds are good that you have dogbane beetles. And you can see just sort of how shiny they are. They look like something that you would find in a tropical rainforest, not necessarily in the fields or road ditches of South Dakota. So I just wanted to share that pretty cool insect with everybody in case you had seen it around and wondered what it was. And I also wanted to share it because it is a little bit of a look-alike with the Japanese beetle. So Japanese beetles, if you're on the I-29 corridor, or especially if you're in the southeast part of the state, Yankton, Vermilion, Sioux Falls, Sioux City area, Japanese beetles are out and they're probably munching on your flowers. This is a glamour shot of a Japanese beetle that I took when I was in Minneapolis at the beginning of July, which seems like ages ago at this point. But they're also a shiny metallic beetle. This one's a little bit more metallic brown, but they're usually more of a metallic green color. And you can see sort of around the edges of the abdomen, you've got that white tufting, which is very characteristic. Instead of having that smooth shell on the back, you can see they've got some ridges on those elytra. But these beetles are really generalist herbivores. There's a list of over 200 different, species of plants that these will eat. So you can't really plant your way out of Japanese beetles. What you can do is work to manage them in their larval stage, because they will eat, their grubs will eat turf grass fruits. So if you're, especially in the southeastern part of the state, if you haven't scouted your lawn for grubs, you should absolutely do that. If you've been noticing some like bare dead patches, kind of peel back the edges of those patches and see if you can see some white grubs in there. And if you've got white grubs, there are some granular treatments that do need to be watered or irrigated in order to be effective. And we're pretty much in the time period where now is when you would want to start treating for those. So this sort of time period as we're moving into August is when the eggs have hatched and the grubs are at their smallest stage. So they're sort of the easiest life stage to kill with chemicals. Once they get to the adult stage, there's not really a whole lot you can do except for pick them off and drown them in soapy water. There are pheromone traps that are available, but those will pretty much just bring all the Japanese beetles to your yard. So I know we've made the joke in some meetings that, you put the trap in your neighbor's yard and they all go over there. But those traps will sort of overflow and then you'll have Japanese beetles eating everything in your yard. So handpick the adults and work on the growth control to sort of manage their populations for future years. I also wanted to throw cicada killers in there because I have gotten so many calls and questions about them. They're, again, having a very good year. Really, you only need to worry if you're a cicada. And so in this picture, you can see the wasp is actually carrying a cicada. So she has paralyzed that cicada and is going to take it back to her nest. These are solitary, so they live sort of alone. You might have a bunch that are using the same area, but they are sort of working individually. They're solitary, they are ground nesting, so they are digging a hole in the ground, and because of their size, those holes can be somewhat sizable, or the dirt mound might be very noticeable. But these adults are going to be active for another couple of weeks, and then they usually are done before summer is over. So they kind of come out with the cicadas, utilize those for their larvae, and then those larvae will eat the cicadas. They will pupate, and then those adult wasps will emerge next summer. So they are huge. I know they look terrifying. They really don't care about people. If they are nesting in a place you don't want them, I recommend sort of managing so that you've got turf or some sort of ground cover there, because they do very much prefer bare ground. So if you're in the Pier area, you go hike down behind the Isaac Walton League, anywhere where we've got sandy trails, you will absolutely be seeing cicada killers. So they just have a really cool life cycle. They're our biggest wasp in the state. They're still not murder hornets, but they are active right now, and we are hearing a lot about them from folks calling our offices. I also wanted to throw in earwigs. I did have a question from a client who thought that earwigs were causing the bark to fall off of their tree. And so I kind of did the deep dive again into earwig biology, and earwigs are decomposers. So they're going to be hanging out in areas of the garden where you've got dampness and moisture. If you pick up your potted plants, they'll probably be underneath the pots outside. I sometimes have them in my milkweed. They do eat living tissue sometimes, but unless you have really high numbers, it's not usually enough to cause a ton of damage. So really managing moisture is the way to go to reduce their numbers, or maybe have them, not be around your pots or things like that. But if you see them on something like a tree, they're more of an indicator of like decomposition. They're not going to be the cause of tree death, and they're generally not the initial cause of plant death. They do have a pretty cool life cycle where the females will actually take care of the larvae or the immature earwigs. So this is an insect that exhibits parental care. So if you've never realized that insects do that, now you know. A little trivia fact about them, and maybe get people to realize that they do some cool things, and it's not just that they're scary looking. And then I did quickly want to share some upcoming events. So if you're somebody who wants to see more insects up close and in person, we will be sort of at Bugapalooza in some capacity. I know the Master Naturalists are going to be there for sure. That is on August 19th in Sioux Falls. If you're in the Huron area, or if you're going to be visiting State Fair, the Fort Building has a whole schedule of events. I'll be there on Thursday and Friday in the morning at like 1 o'clock giving talks on just the 2 days of the fair, but there's great hort exhibits and all sorts of stuff. Thursday is the Value Added Ag Day, so a lot of the specialty crop producers will be in that day event tent. So come see us at State Fair, and you can ask the Master Gardeners questions in person at the Hort Building as well. And then if you're in the Brookings area, we'll be doing Insect Fest on September 9th at McCrory Gardens, and that'll be from 10 to 2. Free day at the garden. So I realized we hadn't been promoting some of our upcoming events, or at least I haven't been on for about a month, but I did want to share those as we're getting closer to some of those dates and also closer to the end of garden hour. So those are the slides that I've got. So I'll go ahead and stop share here. And I'm taking a look at the questions real quick, and there's one
You
about
see
cucumber
the cucumber
beetles.
ones?
Yeah. Oh man. Okay, so you had cucumber beetles, then you used a chemical, and then the spider mites exploded. So here's an interesting thing about why your spider mites exploded, is depending on what you use to kill the cucumber beetles, that also killed the things like lady beetles and some of the other predators that were in there, keeping your spider mite population under control. So one thing that I recommend when folks are scouting their garden and making treatment decisions is don't just count the pests, also count the insects that are helping you sort of do your job in the garden. So pay attention to things like lacewings, to those lady beetles, to syrphid fly larvae, to praying mantids, to spiders, because once you remove those critters, things like, say, spider mites are going to increase in numbers. Spider mites also do really well in hot, dry conditions. So if you are in part of the state, I mean, the whole state is hot right now, but if you're also especially in a place where you're also dry, that is also going to exacerbate spider mite injury. So I'd say, you know, neem is probably going to be fine, especially because it does have sort of more application options than some of the other products out there. But you know, support your plants. Usually squash can overproduce. So really you might have some funky looking leaves, but it might not impact your yield in the long term. And then keep an eye out for those predators the next time before you do an application the next time. So yeah, but interesting question. And yeah, it is pretty much getting close to, you know, leave a zucchini on your neighbor's porch season. So the spider mites might be doing you a favor.
We have another question in the Q&A on garden compost. Madalyn, you might want to weigh in on this one also. My city allows people to get soil from the citywide lawn and garden compost. One doesn't know if there were any infected plants thrown into the yard waste. So is that an issue for virus infected plants? Could we be infecting our yards from city compost? And I don't know if Madalyn stepped out for a minute, but if it's composted correctly, the temperature should be warm enough to inactivate any of those materials. They should be, you know, thoroughly killing any green tissue. And so I think that the likelihood of transferring viruses from compost would be fairly low. Now, if you're just throwing it on your compost pile in the back, unless you're really scientific about it and make sure it reaches 131 degrees Fahrenheit for 3 days or 15 days or whatever it might be, then you do have to worry a little bit more about that. There's one other question there that I am not a turf grass herbicide specialist, but there was a question about brome grass in lawns, and is there selective herbicide for that? And as far as I know, there is not. If you're using a pre-emergence, germination preventer, it will help decrease any new plants from coming up each year. But as far as being selective, as far as I know, there's not one that a homeowner has access to. With that, I'll go ahead and close this out by sharing my screen, and that's not what I wanted to share. Let's try this again. Okay. So to wrap it up, if you're really, really slow at gardening, getting out there, or if you're really gung-ho and wondering what you might still be able to mature yet this year, take a quick look at the location that you're in. For Rapid City, we have about 2 months yet before frost. Some of the areas of the state down near Wagner perhaps, or Yankton, have quite a bit more time yet. But pretty much all the state, except maybe in this northwest corner, can do most of these, and these are the same ones I had on last week or the week before. Green beans may be getting a little iffy for the northwest part of the state, but especially if you're looking at the bush beans with a shorter maturity date, you may be able to get those to mature. It's going to depend a lot on how warm our fall is, not just when the frost date is, but if we get enough heat units for beans. The other ones on this list are all cool season crops, so probably will do okay. Peas, depending where you are in the state, may get frosted before you get too much of a crop, but if you're the gambling type, go for it. I wanted to mention a little bit about raspberries, since I had my first raspberry off one of my plants the other night in my yard. As the raspberry canes finish fruiting, they'll start to die back. And you can leave them until fall or winter and then cut them back. But if you have time to get out there and cut them back now, that opens up the patch a little bit, so you get better airflow through, which does two things. It will help decrease foliar diseases and maybe some cane infecting insects. So that's something that's good to do. And it will also help decrease spotted wing Drosophila, and we'll talk about that in a little bit. I'm a little bit in Amanda's territory here, but just in case you never thought about it before, the difference between raspberries and blackberries: when you pick a raspberry, it leaves that inner core on the plant, whereas with a blackberry, the whole thing will come off together. Just in case you've never actually picked them from the plant. A couple of things we might see with our raspberries, especially where we've gone from sudden changes in weather. Sunburn will show up on raspberries, just has a white bleaching on some of the florets. So kind of a funky looking raspberry there, especially if in drier regions of the state, or drier periods of time, probably because you've got less foliage to protect it at that point. We kind of do a balancing act with raspberries between sun exposure and shade, and I'll talk about that a little bit further. But first of all, I did mention earlier that I was going to talk a little bit about crumbly fruit. And once in a while we get questions about, my fruit just isn't nice like it used to be. And it may be caused by a virus. You may or may not see that many symptoms in the leaves, but the fruit is getting sort of this little round ball instead of a nicely developed drupe. And that can be caused by a virus. These viruses, they used to think it was just one virus, and now I think they're looking at 2 or 3 that may be causing this. And those viruses may be, depending upon the exact type, may be transmitted by aphids or by nematodes, which we already talked about, those microscopic worms in the soil. Or these can actually be transmitted from plant to plant by pollen as well. So, or seed, if the fruit drops and it germinates in the soil, it could be carrying it that way too. So that's one of the reasons why we don't generally encourage people to transplant raspberries from one garden to another, but encourage instead to buy virus-free plants from somewhere, so that you're starting fresh and not picking up any of these possible viruses. And spotted winged Drosophila. So this is that little fruit fly that we've talked about over the past few years. This little fruit fly has the obnoxious ability to actually saw. The female can actually saw into the fruit. Most fruit flies cannot penetrate an intact fruit, but this little creature can. And she plants her little eggs in the fruit, and you go out and you find a piece of fruit that's maybe a little bit soft, and you look at it closer, and there's these tiny little, barely visible white larvae all through the fruit. And it will rot the fruit in about overnight. So this has been an issue with all kinds of fruit. We're still learning more about it as time goes by, but wild fruit, tame fruit may feed on pollen in the spring. So there's been a lot of work with it, and commercial growers have had to up the number of sprays or cover their plants with netting, very fine netting. But one of the studies, some of the studies now have begun to indicate that these flies really kind of prefer a cool, moist area. And so they'll be out, at dawn and sunset, but during the heat of the day, they generally kind of hunker down. They also don't prefer, for the same reason, to be out in the exposed tops of the plants. Instead, they prefer to be hiding under the foliage more. So the better that you prune up your raspberries and get the fruit more exposed to the sun, which is why I talked about the sun scald, you'll have fewer infections in that fruit. And I've found that to be true in my fruit at home. The ones that you see when you walk up because they're really exposed are much less likely to be infected. There's some work done in Wisconsin on using plastic mulch, and they use white, black, and silver reflective. And the silver reflective was because a lot of insects use UV light to sort of orient themselves, and so silver mulch can screw up thrips. They can't quite figure out what's going on. But they found that any of the 3 types reduce the larvae in the fruit, thereby increasing usable yield. And once you get a larvae in the fruit, it's not going to be usable because it rots it so quick. Increase the yield by anywhere from a third to, you know, more over 100%. So it had a fair amount of effect. It wasn't 100%. All these sort of practices that we do can help add protection piece by piece. The other thing with these mulches, it makes it much easier for you to see any dropped fruit, and you should go out there on a daily basis and pick up any dropped fruit, even the yucky, rotten stuff, because that's where those larvae are, and they have a pretty quick life cycle to produce new flies and new larvae. So so that was, use mulch if you can, pick up the fruit, use good pruning practices. And then on top of that, if you want to add insecticides, then make sure you read the label and follow the directions very carefully, particularly paying attention to anything that says PHI or pre-harvest interval for those insecticides. And with that, I'm going to quit sharing. And there was an announcement, State Fair horticulture entries are due by July 31st, and you can enter online. So if you're wanting to share some of your actual fruits of your labor and take it to the state fair, take a look at that and see what you can bring and how to prepare it. And with that, I'd like to thank our panelists tonight, Dr. Shires, Dr. Bachman, Connor, and we hope to see you next week. If you, in the meantime, have questions about anything related to gardening, we have the garden hotline with the email, and the phone number was in the chat box, so you can look at that, or you can go to extension.sdstate.edu, go to the garden yard heading and select problems and solutions. So a number of ways for you to get your questions answered. And with that, I bid you all good night and good week.